Hello everyone, I'm Dr. Monica. Today we'll begin our Periodontology Master Series with one of the most fundamental and important topics in periodontics, that is dental plaque. So let's begin. So before learning the definition, let's understand why dental plaque is so important. So periodontal diseases such as gingivitis and periodontitis, they begin with the plaque accumulation on the tooth surface and adjacent structures. So plaque removal at early stage can prevent these diseases. But if the plaque remains on the healthy tooth for a long period, it can cause gingivitis, which can further progress to periodontitis and eventually lead to tooth loss. So we can conclude that dental plaque, it is the primary etiological factor for periodontal disease. So coming to the definition of dental plaque, so according to Kerenza 11th edition, dental plaque is defined clinically as a structured, resilient, yellowish grayish substance that adheres tenaciously to the intraoral hard surfaces, including removable and fixed restorations. So here the word structured means that plaque is not a random collection of bacteria. It has an organized architecture. Resilient means that it is tough. Yellowish gray substance is suggests the color of the dental plaque. Adheres tenaciously means that it is firmly attached to the tooth surface and it simply cannot be washed away with water. So now let's understand the composition of dental plaque. So dental plaque, it is mainly composed of bacteria, which comprises about 75% of dental plaque. Then we have extracellular polymeric substance matrix, which is composed of 15%. Then we have is host-derived components from saliva and the GCF, which makes up about 7% of dental plaque. And we have only 3% which is made up of inorganic minerals that contribute to the calculus formation. Now let's understand the classification of dental plaque. So mainly dental plaque, it is of three types, that is supragingival, subgingival and tooth associated cervical plaque. So the supragingival plaque, it means that the plaque which is present above the marginal gingiva. So mainly this plaque comprises of cocci dominated gram positive bacteria which are mainly aerobic in nature. Next is the subgingival plaque. It means that the plaque which is present below the marginal gingiva and it is mainly located in the periodontal pocket or within the gingival sulcus. So mainly it is composed of anaerobic gram-negative motile bacteria. So clinical importance of subgingival plaque is that it is associated with attachment loss which can lead to bone loss and cause periodontitis. Third type, it is tooth-associated cervical plaque, which is mainly located near the cemento-enamel junction, that is the enamel surface just below the gingival margin. So it is the transition zone flora, that is we can see mainly gram-negative facultative anaerobes. The clinical importance is that it is mainly responsible for the initiation of root caries and it can also aid into the gingivitis and pocket depth progression. So let's understand what dental plaque is. So dental plaque is a biofilm, that is, it is a community of bacteria embedded in a self-produced matrix that sticks to the tooth surface. So why the bacteria prefer to live in this biofilm? So firstly, it protects the bacteria from host defenses and environmental stresses. Secondly, it is easy for the bacteria to communicate and share nutrients in this biofilm. Also, due to this biofilm, the plaque, it is resistant to the antibiotic. Also, the bacteria becomes dormant and it can survive the antibiotic treatment. So let's understand this biofilm with a simple example. Just imagine that the free floating bacteria, they are like these individual people. So coming to the biofilm, it will be like a well organized city where inside a city, the bacteria, they communicate, share nutrients, and protect each other. That is, in this example, they are the individual people. So now let's understand the most important part of dental plaque, that is its formation. So it is formed in three stages. That is, first stage it is the pellicle formation, second is initial adhesion attachment, third is colonization or the plaque maturation stage. So let's first understand the first stage of dental plaque formation, that is pellicle formation. So whenever we eat something, just within few seconds, the proteins and the salivary glycoproteins present in the saliva, such as the stearin, mucin, proline-rich proteins, they begin to get absorbed onto the enamel surface, and these molecules they bind tightly to the hydroxyapatite crystals of the enamel, and they form a very thin acellular protein rich layer, and this layer it is called the acquired pellicle. So at this stage the pellicle it contains no bacteria. It acts as a protective conditioning film, and the main function of this acquired pellicle is to prepare the tooth surface for the next stage where the bacteria begin to attach. So coming to the next stage, once the acquired pellicle has formed, the next stage is called the initial bacterial adhesion or attachment. This usually begins within few hours after the pellicle formation. So the first bacteria, they are the primary colonizers which get attached. These are mainly the streptococci and the actinomyces. And these bacteria, they possess molecules called the bacterial adhesins, while the acquired pellicle, they contain complementary receptors. So when these adhesins, they recognize and bind to these receptors, the bacteria, it gets attached to the tooth surface. So initially the attachment of the bacteria to the tooth surface, it is weak and it can be reversible, but later on within a short period of time the interaction it becomes stronger and irreversible, which allows the bacteria to remain firmly attached to the tooth surface. So these primary colonizers they are very important because they modify the tooth surface and create suitable environment for the other bacteria to attach during the next stage of plaque development. So coming to the third stage, that is colonization and plaque maturation. So after the primary colonizers they have attached, secondary colonizers they begin to join through a process called coaggregation. So as more bacteria they accumulate, they produce the extracellular polymeric substance which acts as a protective glue, and they hold the biofilm together. So over the next few days to weeks, the plaque it will become thicker and it will become more organized. So we can see there are fluid channels. They are developed to transport the nutrients and also to remove the waste, while the oxygen levels it decrease towards the deeper layer. So we can see that in the deeper layer there are highly anaerobic bacteria, and in the middle we can see facultative microorganisms, and in the outer zone they are usually anaerobic. So in the deeper layer, we can see that mainly the Porphyromonas gingivalis, and they are usually able to thrive. So at this stage, a mature dental plaque biofilm is formed. So now let's quickly understand how the dental plaque it develops into a mature biofilm through colonization and co-aggregation. So in the stage 1 we can see that the primary colonizers such as the gram-positive Streptococcus, Actinomyces species and Veillonella species, they are the first microorganisms. They get attached to the acquired pellicle and they establish initial bacterial community. Next, we have is the co-aggregation. That is, it is a process by which the different bacterial species they attach to one another through a specific interbacterial addition. That the main key organism in this stage is the Fusobacterium nucleatum, which acts as a bridge species. So it connects the early colonizers with the late colonizers, which allow the biofilm to grow in complexity. Next is the third stage. In this, the secondary colonizers such as the P. gingivalis, P. intermedia, T. forsythia, and T. denticola, they join the biofilm. As these bacteria accumulate, they produce the EPS, and that binds the microorganisms together as a glue and forms a well organized three-dimensional biofilm. So this biofilm, it contains heterogeneous microcolonies, and the deepest layer, it is mainly composed of anaerobic, creating an ideal environment for the periodontal pathogens. So this mature biofilm, it is more resistant to antimicrobial agents and host defense mechanisms, which makes it a major etiological factor for periodontal disease. So the main important thing here is the Cob formation. In this, we can see that in this arrangement, a central filamentous bacterium, mainly the fusobacterium or actinomyces, can be surrounded by cocci, which is mainly the streptococcus, since it gives the appearance of corn cob. So it is a characteristic feature of a mature supragingival dental plaque biofilm. So this is a mature dental plaque biofilm. So now let's understand the metabolism of dental plaque biofilm. So one important feature of a biofilm is that the bacteria do not live independently. They cooperate metabolically. In simple words, the waste product of one bacterium becomes a nutrient for the another bacterium. And this is called metabolic cooperation or cross-feeding. So during the early stage, the primary colonizers such as the Streptococcus species, the Actinomyces, and the Venolena species, they metabolize the dietary sugar and they produce metabolites such as the lactate and fumate. And instead of being wasted, these metabolites, they are utilized by other bacteria within the biofilm. As the plaque matures, the late colonizers such as the Porphyromonas gingivalis, P. intermedia, and Fusobacterium nucleatum and Treponema denticola, they utilize these metabolic products and they break down the proteins from the GCF. So this results in the production of substances such as the butyrate and ammonia, hydrogen sulfide, cadaverine, and putrescine. And these metabolic products, they help maintain the biofilm, which create an anaerobic environment, and they contribute to the oral malodor and can damage the surrounding periodontal tissues. Therefore, the metabolism of dental plaque, it is not the single bacterium activity, but it is a coordinated interaction um, with among the many bacterial species. So, the supragingival plaque formation depends on many factors such as the salivary factors, that is the rate of salivary flow, salivary composition, the acquired pellicle, the tooth factors, that is the morphology of the tooth, surface roughness, patent fissure, and presence of any restoration. The microbial factors, which include the early colonizers, bacterial adhesion, and the biofilm maturation. Dietary factors, which include sucrose intake, frequency of food, and sticky food. Environmental factors that include smoking, orthodontic appliance, xerostomia, oral hygiene that includes brushing, flossing, and interdental aid use. So coming to the methods of plaque control, we have mechanical plaque control methods, which can be done by using brushing, use of floss, and use of interdental brushes. Coming to the chemical plaque control, in this mainly we have use of mouthwash and use of toothpaste. And in the professional care, we can do scaling and root planing and the maintenance after these procedures. So next we have is the plaque hypothesis, which we will discuss one by one. So coming to the first plaque hypothesis, that is the non-specific plaque hypothesis. According to this, periodontal disease, it is caused by the noxious products released by the entire plaque flora. So coming to the second plaque hypothesis, that is the specific plaque hypothesis. According to this, Sokransky's red, green and orange complex bacteria, they are associated with the periodontal disease. Next is the updated plaque hypothesis. According to these, any bacterial colonization, that is the plaque of sufficient quantity in the gingival crevice, always causes at least gingivitis. So next is the ecological plaque hypothesis. According to this, the total amount of dental plaque and specific microbial composition of the plaque, they contribute to the transition from health to disease. So the next hypothesis is the keystone pathogen hypothesis. So in this, the certain low abundant microorganisms, they can cause periodontitis by changing the composition and increasing the quantity or the number of the normal microorganisms. If you have liked the content, please like, share and do subscribe to this channel. Thank you.